Understanding the impact of assisted hatching, timing on embryo. Genetic testing in IVF-assisted hatching is a common laboratory technique in IVF, where a small hole is made in the embryo's outer shell to help it implant. There's been ongoing debate about whether it's better to do this early, days 2 to 4, or later, days 5 to 7, especially since embryos are often tested for genetic abnormalities, PGTA, before being chosen for transfer. Some have worried that the timing of this step could affect the results of genetic testing, particularly the rate of mosaicism, when an embryo has both normal and abnormal cells. This large retrospective study analyzed 1,260 embryos from 134 clinics, all processed at a single genetic reference laboratory. The embryos were split into two groups, those that had assisted hatching early and those that had it late, either before or at the time of the biopsy for genetic testing. The researchers looked at key outcomes: the rates of mosaicism, euploidy, normal chromosome number, aneuploidy, abnormal chromosome number, and the proportion of embryos where no result could be obtained. The main finding was reassuring. The timing of assisted hatching made no significant difference to any of these outcomes. Mosaicism rates were similar for both groups, about 15%, and there were no significant differences in the rates of normal or abnormal embryos. This remained true even after adjusting for important factors like the age of the patient, the day of biopsy, and the method used to amplify DNA for testing. Even when embryo quality was considered, the results did not change. In practical terms, this means clinics and embryologists can choose when to perform assisted hatching based on what works best for their workflow and technical preferences, without worrying about negatively impacting the genetic health of embryos. This flexibility can make IVF more efficient and less stressful for both staff and patients. Overall, this research provides strong evidence that the timing of assisted hatching does not influence the rates of mosaicism or other genetic outcomes in embryos tested with PGTA, does not affect their chances of success. Reference Lee It, McWilliams K. Berger DS. Timing of assisted hatching and mosaicism rates after trophectoderm biopsy for pre-implantation genetic testing for aneuploidy. Fertile Sterile, 2025, doi 10116J Fertenstert 225 0100.